Hey guys, it's Mrs. Olenichek, and today we're going to go over the diffusion through a membrane lab. So if you remember, the first step was to fill some dialysis tubing with a glucose and starch solution. The dialysis tubing represents a cell membrane or a selectively permeable membrane. It has tiny pores in it, and so it will let certain substances pass through. You then placed your artificial cell, that dialysis tubing with the starch and glucose, into a cup or a beaker and filled it with water and added Lugol's iodine till it turned an amber color. That was your initial setup for the lab. So Lugol's is a starch indicator, and so it'll change color in the presence of starch. At this point, you should write down your predictions for what's going to happen in your table. So you should have written down your initial setup. You had inside of the bag, you have starch and glucose, and outside you have water and iodine. Then you can make some predictions about what you think is going to happen and why. While you were waiting for that to diffuse, or not to diffuse, you went ahead and did some nutrient testing. So we want to know what a positive test result looks like, and we want to know what a negative test result looks like. So that way later on you can compare your results and figure out what happened. So you'll notice here, this is the starch test with Lugol's iodine. So you added some iodine to 3 solutions. One was glucose, one was starch, and one was water. So glucose plus iodine and water plus iodine stayed that amber color, and so that's what a negative test looks like. But when you combined the iodine with starch, it turns that blue-black color, so that was a positive test result. The glucose test is similar, where you had water plus a Benedict solution. That was that really pretty blue indicator, and so we combined that with water, glucose, and starch in 3 separate test tubes, but this one you had to heat up. And you can see here, when you heated it up, the glucose plus Benedicts turned to an orangish kind of color. After about 30 minutes, if you come back to your cell, it should look like this. And so now we see that inside changed a blue-black color, and that tells us that the iodine diffused into the cell and reacted with the starch. And this also tells us that the starch did not diffuse out of the cell. In order to figure out whether the glucose was able to diffuse, we had to test that outside area. So we tested out here. And you can see over here what our results were. So we ended up with a positive result outside. So that tells us that afterwards, inside of the cell, we have starch, glucose, because it's not going anywhere. At least not all of it would diffuse out. And iodine, and we know the iodine's there because it turned that blue-black color. And then outside, we know we still have water and iodine, and we tested to figure out whether there was glucose, and so we see that glucose did diffuse out. Now if you think about the structure of starch and glucose, glucose is a simple sugar, so it's a monosaccharide, so it's a small molecule, whereas starch is a polysaccharide, so it's made up of lots and lots of monosaccharides, so it's a much larger molecule. So it makes sense that it wouldn't be able to diffuse through that type of membrane. So here's what your table number 2 should look like. You should have your predictions in there too. Take a minute, check over yours, make sure that you filled in everything you needed. All right, now I'm going to go over some of the summary questions with you. So first we wanted to know, did glucose diffuse out of the cell? Explain how you can tell. Well, yes, glucose diffused out of the cell, and this was confirmed by a positive Benedict's test. So when we tested the solution outside of the cell, we noticed that when heated with Benedict's, it turned from blue to an orange color, which confirms that glucose diffused through. Did the starch diffuse out of the cell? No. If it had diffused out of the cell, then we would expect the outside solution to have changed blue-black, but it did not. And the color change that occurred inside of the cell, Well, that was because iodine did diffuse through the cell membrane. So iodine's a small molecule as well, so it was able to fit through the pores. It went from where it was in a high concentration outside of the cell to where it was in lower concentration inside of the cell and reacted with the starch. So remember, all of these are examples of diffusion, molecules going from high to low. All right, so number 4, based on your test, which substances diffuse through the dialysis cell membrane? So we tested glucose and we saw iodine tests. So those are the two that we know for sure passed through that membrane because we tested for them or we saw a color change. We also know that starch did not diffuse through the cell membrane because there was no color change outside. And the whole reason for this is going to be that glucose and iodine are small molecules that can fit through the pores in the membrane, but starch is a large molecule that couldn't pass. So because those pores were of differing sizes, only some of the molecules were able to pass through. So we had two basic variables going on in this experiment. You had size of molecule and you had concentration. And so this is looking at the size of molecule and how that impacted it. We didn't measure it, but we can assume that water also passed through that membrane. We had a much higher concentration of water outside than inside, and so we assume that it passed through the membrane. We could measure the cell before and after. We could look at its volume or its mass, or we could look at the volume before and after of the outside environment of the cell to try to measure whether or not water has passed through or how much water would have passed through. So glucose diffused out of the cell because there was a low concentration of glucose outside of the cell and it was trying to reach equilibrium. So remember, whenever possible, substances are going to diffuse from an area of high concentration to an area of low concentration, and they're going to continue to do this until they reach equilibrium. At that point, there's still going to be movement back and forth, but there's going to be no net flow. So the glucose diffused out because there was a lower concentration outside of the cell. And the iodine flowed into the cell for the exact same reason. There was more iodine outside, so that's where you had your high concentration, and less iodine or no iodine inside of the cell. And so because it could go through the pores, it was able to flow into the cell to reach equilibrium. All right, so what if you set this up wrong? Let's think about that. If you put water and iodine inside of the dialysis bag and starch and glucose outside, you'd get the same kind of result, but you'd notice that the starch is going to stay outside of the cell with glucose diffusing into the cell. The iodine would still diffuse to where there was a lower concentration, in this case outside of the cell. And so the cell itself would be amber colored and the outside environment would be a blue-black color. All right, here's the big question. Why is this important? What does this have to do with you? So animals, including humans, often eat starches in the form of fruits and vegetables, breads and baked goods. Remember, we talked about complex carbohydrates. The starches are digested into glucose in the small intestine. One of the enzymes that does this is called amylase. The glucose then can diffuse through the cells in the small intestine to the bloodstream and then to the cells that need it. And remember, we use sugars for short-term energy. So it makes sense for a cell or an organism to break down starch into glucose so that it can easily diffuse through the membrane. You don't want to waste a lot of energy transporting something so that you can get energy. And so it's more efficient to break it down, have it diffuse down its concentration gradient into the bloodstream, and then diffuse into the cells that need it and be able to go through the membrane without having to pump it through or without having to form a vesicle for something as large as starch. So by digesting the starch into glucose, you can make it easier to transport across the cell membrane from an area of high concentration to an area of low concentration without needing to input energy to form a vesicle for a large molecule like starch. So that's it for this lab. Go back, check over your answers, add everything up, and then the next video will show you the osmosis lab. All right, have a good day.